Today we are looking at genetics. So we are going to look at part 1 of genetics, that is introduction to genetics. We are going to discuss in another lesson further on genetics. So today, as we start with the genetics, first we have to define, to give a definition of genetics. So what is genetics? Genetics, it is a branch of science or it's a branch of biology that deals with inheritance. It deals with inheritance and variation. So, genetics is a branch of science that deals with inheritance and variation. So, inheritance. So, before we come to any other thing, what are we talking about inheritance and what is variation? So, inheritance, by definition, inheritance, it is the ability to transfer genetic traits from parents to offspring and change. So inheritance is the ability to transfer genetic information from parents to offspring and change. That is what we call inheritance. Now when we talk about variation, it is the difference in characteristics of a particular trait. That is what we call variation. The difference in characteristics for a particular trait. For example, when you are having a father who is tall and marries maybe a wife who is dwarf, the offspring of these two people, either the child is going to be tall or dwarf, depending on genetics or depending on inheritance, which you're going to look at further stages of genetics. We're coming to that point. Now, in short, what we are saying is this. If an African marries an African, the child is also an African. That is black pigmented skin. If a white or a European marries a European, the child is going to inherit that blending of the color of the European. So therefore, inheritance is that ability to transfer genetic traits from parent to offspring and change, and variation, it is the difference in characteristics. The characteristics we are talking about, for example, in a family, you will find that some children are tall while others are short. Others have got light skins, others have got dark skin. So when you talk about color, we are not the same. So there's a difference in our color blending. So that's what you're talking about. That's what we call now variation. So genetics, it is the study of inheritance and variation, where inheritance is the ability to transfer genetic information from parents to offspring and change. Then variation, it is the difference in characteristics of a particular trait in that matter. Then, after definition of inheritance and variation, we have types of variation. Now, there are two types of variation. One, we have continuous variation and we have discontinuous variation. What is the difference between continuous variation and discontinuous variation? Let's start with continuous variation. Now, in continuous variation, it is a phenomenon or is a state whereby there is a wide range in difference in characteristics of a particular trait. There is a wide range in different characteristics of a trait. For example, color, height, mass, fingerprint, etc. What do I mean by this? That tells us that in a class of 40 students, and you're supposed to determine their height, we cannot group them in terms of, let's say, those that are 1 meter, 1.5 meter. No. These students, in that class, we are going to have different heights because each and every person have got their own heights. When we talk about color, we are all different. Even if you are African or black, our blending is not the same. There is difference in that color in our skin, even in our family. When we talk about mass, we don't have the same mass. That mass keep on changing from one student to another, and that is now what you're calling continuous. There is a wide range of that. So if you have to draw a graph of mass, you're going to have a curve, not a straight line. You're going to have a curve where others are more, others are less, and such. So therefore, when you're talking about continuous variation, by definition, it's a kind of variation where there is a wide range of difference in characteristics of a particular trait. So that is continuous variation. Then, when we talk about discontinuous variation, now what's the difference between continuous and discontinuous? Now, in discontinuous variation, this one, there is a distinct, is a kind of variation where there is a distinct difference between one trait, between a characteristics of a trait and another. So what do I mean? The word distinct. You are going to be able to distinguish between this and that. For example, gender. In a class of 40 students, being a mixture of boys and girls, it is very easy to say, let the boys be on one side and the girls be on the other side. So you are either male or female. You are either male or female. Again, you can also talk about another example, that is blood group. Again, in the same class, we can locate the student by saying, let the student of blood group A, AB, B, and O group themselves. So you find that it is easy to have a group of blood A, a group of blood group B, a group of blood group AB, and a group of blood group O. So these two examples, we are able to distinguish boys and girls according to physical characteristics and such. When we come to blood group, we can also do that. So that is an example of a discontinuous what? Variation. Then we have another example, that is rolling of tongue. There are people who can roll the tongue and others who cannot. So we can able to differentiate between those that are tongue rollers and those that are non-tongue rollers. So there are those who can and those who cannot do that. So therefore, gender, blood group, and tongue rolling, let them be an example of discontinuous variation. So we have now the two types of variation, that is continuous variation and discontinuous variation. Again, what causes this variation? What is the cause of variation? What are the causes of variation? Now, we are going to list them and then we are going to explain them one by one. Now, one cause of variation, it is fertilization. Fertilization. Number two, we have crossing over. Then we have independent assortment. And we have mutation. So those are the main causes of variation. Now let's start with fertilization. Now what is fertilization? What do you understand by fertilization? Now fertilization, it is the fusion of the male gamete and female gamete. Fertilization is the fusion of the male gamete and the female gamete. That is the nucleus of the male gamete and the nucleus of the female gamete. For human beings, it is the fusion of the sperm cell and ovum to form a diploid zygote. The moment the sperm cell fuses with the ovum of a female, then they form a diploid zygote. That is what we call fertilization. Now the question is, how does fertilization cause variation? Now you understand that in a human being, we have got 46 chromosomes, or let's say we have got 23 pairs of chromosomes. Now, a sperm cell is a haploid cell, and an ovum is also a haploid cell. What does that mean? A sperm cell have got half number of chromosomes, and the ovum have got half number of chromosomes. When they fuse together, they form now 46 pair of chromosomes of a human being. But how is fertilization causing the variation we are talking about? This because the sperm cell carries the genetic characteristics of the father, while the ovum carries genetic information of the mother. Now, when the sperm cell and the ovum fuse, forming a diploid zygote, that zygote comprised of a mixture of characteristics of the father and characteristics of what? The mother. So you realize that the offspring or the child that will come out from there is going to have some characteristics of the father and some characteristics of the mother. So the child is a blending of the two people, or the two organisms, to form blended characteristics. That means that the child cannot look exactly like the mother and cannot look exactly like the father, because the child comprises of two organisms with different characteristics that fuse together to form one organism. So that is how fertilization causes variation. Now, when we talk about number 2, that is crossing over. Now, crossing over deals with cell division. Now, during cell division, you realize that at some stages of cell division, maybe prophase, you understand that in the cell, the chromosomes that are scattered inside the nucleus, inside the cell, the chromatids may come into contact with one another at a point called chiasmata. Now, the point at which these two chromatids come into contact, that point is called chiasmata. You realize that the genes or the characteristics are located in the chromatids in position called gene locus. So therefore, when the two chromatids come into contact with one another, the genes or the characteristics might cross over from one chromatid to another and another from this side to this one. So when the two chromatids separate, this chromatid is going to have some characteristics of the other chromatid, which happens also to this one when they are separated. So if this chromatid happens to give an organism after cell division, that organism is going to have different characteristics because there was an exchange of information or genetic information at the point of contact. So that exchange of genetic information at the point of contact will result to variation at the end of it all. So that is how crossing over causes variation. We also have independent assortment. Again, during cell division, we have got stages of cell division, that is interphase, prophase, metaphase, anaphase, and telophase. During cell division, as the chromatids and the chromosomes align themselves at the equator, as they move to opposite poles of the cell, they do it independently. Assuming this cell was comprising of, chromatids of a mother and their father. That means these chromosomes that we are seeing inside here is a mixture of, let's say, father, mother, and such. So when the chromatids are moving in opposite direction, they do it independently. So as they do it independently, you're going to realize on the upper side you're going to have a mixture of chromatids from the father and chromatids from the mother. The same thing, the chromosomes down here are going to be that of the father and that of the mother. So that tells you that after these cells have divided into two cells, after cell division, this cell is going to have a mixture of two organisms. And this is going to have a mixture of two organisms as well. So that also is going to cause variation of the offspring. So that is how independent assortment causes variation. Then we have mutation. How does mutation cause variation? So first we have to define or to give a definition for mutation. Now mutation, it is a spontaneous change of genetic constitution of organism. It is the spontaneous change in genetic constitution of organism due to exposure to mutagens or to things that cause mutation, such as radiation, such as chemicals and heavy metals and such. So example, you can have a man and a woman. In the family tree of this man, there has never been a case of an albino. And on the other side, on the family tree of a woman, there has never been a case of a child who is albino. But when these two marry and they give birth, the wife gives birth, you realize that in rare cases, they might give out an albino. Now, you realize this man has no history of albino, the woman has no history of albino, but now they have an albino child. What does this mean now? This means this organism that's an albino is what we call now a mutant. And the phenomenon is mutation. It is mutation that causes this. So later in the topic, you're going to look at mutation as a topic, what causes mutation, and the types of mutation. But by definition, mutation is a spontaneous change in genetic constitution of organism. And that is the example. So after you've seen genetics, definition of genetics, inheritance, variation, type of variation, and causes of variation, we are going to look at the structure and function of DNA. The structure and function of a DNA. So what is a DNA? DNA is a protein molecule called deoxyribonucleic acid. It is deoxyribonucleic acid. Now, a DNA, it is the protein molecule that contains genetic constitution of any organism. It is a protein molecule. It is a protein molecule that contains genetic constitution of organism. That is what we call a DNA. Now, before we look at the function of a DNA, let us look at the structure of a DNA. Now, in terms of structure, DNA comprises of three main parts. One, it comprises of a phosphate molecule. Number two, it has what we call nitrogenous base. which comprise of thymine, adenine, cytosine, and guanine. Then number 3, we have the carbon sugars. Carbon sugars. So by structure, DNA comprises of phosphate molecule, nitrogenous base that comprise of thymine, adenine, cytosine, and guanine, and then we have the carbon sugars. So let us draw that DNA and be able to differentiate between the phosphate molecule and the nitrogenous base and carbon sugars. So we have a strand here, which we call now phosphate molecule, and connected by carbon sugars and a nitrogenous base at the far end. Again, carbon sugars and nitrogenous base, carbon sugar and nitrogenous base. So we have got double strand. It is two-sided strand. It has got two strands. So that is the structure of a DNA. Now, this thread here is phosphate molecule, which is also on the other side as a phosphate what? Molecule. Then we have the carbon sugars. Then we have nitrogenous base. Nitrogenous base. Now, nitrogenous base comprises of thymine, adenine, guanine, cytosine, thymine. It keeps on changing like that. Now, the thymine articulates with adenine. So, thymine, adenine, thymine, guanine, cytosine, guanine, thymine, adenine. So, they alternate like that. We have the thymine goes with adenine, adenine goes with thymine, guanine with cytosine, cytosine with guanine. So this is basically the structure of a DNA. Now, this strand of one unit of one side of a DNA, we call it a nucleotide. That is DNA nucleotide, one part. But when they are two combined, we call it double helix strand. A double helix strand. So that is basically now the structure of a DNA comprised of phosphate molecule, carbon sugars, and nitrogenous base at the center, comprising of thymine, adenine, guanine, and cytosine. So after the structure of the DNA, we look at the functions of the DNA. So, what are the main functions of the DNA? Now, functions of DNA are: 1, it is able to transfer genetic information from parents to offspring unchanged. Able to transfer genetic information from parents to offspring unchanged. So that is the first function of DNA, that is able to transfer genetic information from parents to offspring unchanged. Number 2, it is able to translate genetic information into characteristic of organism. It is able to translate genetic information into characteristic of organism. And number 3, it is able to store genetic information in coded form. So we have got these three main functions of the DNA. Transfer genetic information from parent to offspring unchanged, And number two, translate genetic information into characteristics of organism. And number three, it is able to store genetic information in coded form. So that is the structure of the DNA and the function of the DNA. So for today, let's put it there. The next lesson will continue from there. Today we just looked at the introduction of genetics.